So I'm Martina Boiardi. I am from Italy. I studied biomedical engineering. And then I studied in Polytechnic of Milan, and then I decided to start a PhD, and I joined the Helens Lab at KU Leuven. And actually it's a joint PhD between the Helens Lab and the Lambertin Lab. I study the cell cycle. This is like the short explanation. I work the Xenopus levis, with Xenopus levis frog. In particular, we use the eggs of these frogs. So it's particularly convenient because at the early embryonic developmental stage, the cell cycle is very fast. It's not like the normal somatic cell cycle, which can be quite long, and there are the gap phases and the checkpoint, but instead you just have a cell that oscillates between interphase and mitosis, and the cells within the embryo, they keep dividing without the embryo growing in size. So you can use the in vivo model per se, or you can take the eggs, and from the eggs you can make extract. Basically, you just pull them together, you crush them with an ultracentrifuge, and then you extract the cytoplasm. And in the cytoplasm, you still have everything that you need to do cell cycles. So basically, there are still the proteins that regulate the cell cycle in the normal embryo, they are still present. And within the extract, you can observe cell cycle oscillations. Of course, it's not real cell cycles. It's a simplified model to study certain mechanisms. And in particular, what I am looking at is the interaction between the cell cycle oscillator, which is like the main proteins are cyclin B and cyclin-dependent kinase 1, and how these interacts with microtubules and in general cytoskeleton, but I'm more looking at microtubules organization, because we think that there might be a link, that microtubules and more in general self-organization of the cytoplasm can also have an effect in regulating the speed at which the cell cycle. Okay. So I started my PhD I think 2 years ago. It's challenging. I was particularly so maybe one thing that I should also say is that I do not only work with the extract per se, but we encapsulate this extract in droplets and to make like kind of artificial cells. And we observe the dynamics within these cells. So what I'm mostly doing is working either with these simple cells like that, or building microfluidic devices to build these cells. So I would say that I have two main exciting results to date, because first of all, regarding the microfluidics, I'm now able to make droplets with a controlled size. Which is very interesting, because then you can try to understand if certain mechanisms have like a way of behaving in a smaller or bigger droplets, and then also trapping these cells, these artificial cells on chip to be able to image them. This is good because otherwise, what I normally do is just encapsulating these droplets in glass slides, and then in these glass slides, so they're like tiny capillaries, 100 micrometer high, they can move, they can merge, so that's very difficult then to do the analysis. But then if you can trap the droplets into traps on chip, then it's really nice. So that's one thing that I'm really happy about, because I'm finally doing it. And another very interesting result is that we actually observed that we think that it's the way to go, that depending on the level of organization of the cytoplasm, we observe a different speed in the cell cycle. So if we only have microtubules, we see that things happen relatively faster. If we add components, like for example centrosomes, so we isolate them from extract with other techniques, and then we add them to the cycling extract, we see that the cell cycle starts slower, and then it also slows down during time. And then if you add things that make the cytoplasm even more organized, like nuclei, things are even slower. They start slower and they slow down. So I think it's interesting to try to understand also why this is happening. So this conference, so it's only the second day, but I have to say that one thing that is really catching my attention is actually not my field, but the other fields. So it's really nice to see that there are so many people, so with so many different backgrounds, and what's really interesting is in particular the focus on the environment and the climate change, because we all know that this is happening, but this is the first time for me that I'm hearing like real scientists talking about it. So I'm really liking this. It's also giving me more information and things that I didn't know. I also really like the early career researcher session. It was a good opportunity to try to understand also what I want to do in the future.